Hello everyone, and welcome to today's webinar on AI Microscopy Symposium 8th edition. My name is Wonyoung Choi. I'm the product manager, product owner for Avia's AI image analysis software by Leica Microsystems, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Leica Microsystems. To learn more, visit www.leica-microsystems.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box that you see on your screen and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speakers. Our first speaker is Dr. Helen Spiers, Biomedical Research Lead of the Zooniverse and CZI Imaging Scientist Fellow. Based at the Francis Crick Institute in London, Dr. Spiers works with many research teams around the world to develop novel citizen science projects tackling a large range of biomedical research questions. Dr. Spiers also studies the phenomenon of citizen science and is currently examining multiple aspects of volunteer experience across the Zooniverse. Prior to her current position, Dr. Spiers completed a PhD degree in developmental epigenetics at King's College London and works as a medical writer for 2 years, and obtained a degree in biochemistry from the University of Oxford. Our second speaker is Dr. Martin Priessner, postdoctoral researcher from Astrazeneca in Gothenburg, Sweden. In his PhD, Dr. Priessner was working on strategies for improving microscopy frame rates using machine learning techniques, and he developed fluorescent copper probes for the detection of copper in cells. He's a chemist by training with a degree from Vienna University of Technology and obtained another master's in Neurotechnology from Imperial College London. His research interest is in applying AI solutions to scientific problems to improve measurement techniques and automate the analysis of data. Dr. Spiers, you may now begin your presentation.
Lovely, thank you. So thank you for that kind introduction, and many thanks for the invitation to present today. And thank you all in our audience for taking the time out of your schedules to join us. So as you'll have heard, my name is Dr. Helen Spiers, and I'm a Zuckerberg imaging scientist. And today I would like to tell you a little bit about how we've been applying citizen science for the segmentation of electron microscopy data. And just to apologize, I have a streaming cold, so if I need to go on mute to sneeze or cough, I'll do so. So in terms of what I'd like to cover in today's presentation, firstly, I'd like to give you all a brief introduction to citizen science, because I'm not assuming that any of you will have heard of citizen science previously. I'll then provide an introduction to the world's largest platform for online citizen science, the Zooniverse platform. I'll then go on to describe how we're using the Zooniverse platform at the Francis Crick Institute. And then if there's time at the end of my presentation, I'll give a very brief introduction to how you can build and launch your own citizen science project. So firstly, what is citizen science? So citizen science can be broadly defined as involving the public in research. There are a number of related terms to citizen science, such as participatory research, community-based research, and other terms. These terms have their own specific histories and communities and practice, but they all relate to citizen science. Together, these practices encompass a huge range of different citizen science projects, and the logos here just show a very small fraction of the total projects out there. There are hundreds of citizen science projects out there. So despite their variety, citizen science projects frequently have similar motivations at their core. So central to most citizen science projects is the aim to enable the generation of new knowledge that would not be possible or easy to generate otherwise. Other common motivations behind citizen science projects include improving engagement with science and also democratizing science through improving access to the means of knowledge production, and also allowing members of the public to contribute to asking different sorts of research questions. So via what mechanisms do citizen science projects generate new knowledge? So just to introduce you to some of the different citizen science projects that are out there. Some projects use volunteers' familiarity with their local environment to gather new data. So one well-known project in this context is eBird and Common Sense. So eBird asks birders to share bird sightings via an app, and Common Sense allows members of the public to contribute air quality readings to advance research. There are also projects that ask members of the public to donate idle computer time. And BOINC is one very well-known volunteer computing platform in this context. And BOINC has played host to a huge variety of different citizen science projects, including SETI at Home, which some of you may have heard of. It's a very famous project launched in the 1990s to allow members of the public to donate their idle computer processing time to analyze radio signals in search for extraterrestrial intelligence. And some of you may have heard of Rosetta at Home, which was a project looking to advance understanding of protein structure. There are also projects which ask volunteers to share personal qualitative and quantitative data for further analysis, with well-known examples including Patients Like Me, which is an online platform for quantitative personal research where patients with illnesses can share self-reported outcomes, connect with other communities and learn from pre-existing data. And more topically, more recently, there's also the Zoe COVID symptoms study, which was a mobile app developed in collaboration between King's College London and Guy's St Thomas to collect information in conjunction with COVID symptoms. There are also maker movements in which members of the public connect with other communities to perform wet lab experiments, to work with equipment and to learn protocols and develop skills. And there are also projects that involve the analysis of data. So a well-known example includes Fold It, which was an online puzzle game about protein folding, which members of the public worked to look at protein structure, and then the highest scored proteins were then further analysed by researchers. And it is the Zooniverse platform that falls into this mode of knowledge production, the analysis of data. So the vast majority of projects on the Zooniverse platform involve the analysis of pre-existing data sets. So this brings me on to the second part of my talk. What is the Zooniverse platform? So the Zooniverse is the world's largest platform for online citizen science. So to give you a bit of background to where the Zooniverse came from, so I'm sure most of you here would agree that we're living in an era of unprecedented data production. So although we're moving to a world in which a large proportion of this data can be categorized by computers, ever more so, a significant proportion of this data still requires human pattern recognition abilities for analysis, so to provide the metadata to enable downstream analysis. However, sometimes due to the size of the data set produced or its complexity, it can be impossible or challenging for research teams to do this analysis on their own. And this is exactly the position that Zooniverse co-founder Chris Lintott and one of his colleagues, Kevin, found themselves in now over 10 years ago. And they were working to analyze hundreds of thousands of images of galaxies that had been produced by the Sloan Digital Sky Survey telescope. At the time, although there were computer processing methods for analyzing data, they weren't adequate for providing the metadata that was necessary to analyze the images being produced by the telescope. And so human effort remained necessary to categorize these images. So after manually categorizing tens of thousands of these galaxies, Kevin and Chris calculated that even if they worked 24 hours a day for 7 days a week, it would still take them 3 to 5 years approximately to fully categorize the data set. And so being inspired by similar projects, or some citizen science projects at the time. So Stardust at Home was a citizen science project that had been launched in previous years. They were inspired by the beginnings of citizen science to develop the first Zooniverse project, Galaxy Zoo. And this was a project that simply asked volunteers to look at images of galaxies and to provide some basic morphological information. And after building and launching this project, it was found that this was a highly effective means of generating the data that they needed for their downstream analysis, and it was incredibly popular. So in the first few hours after launch, the project received tens of thousands of classifications per hour. And it was found that because multiple volunteers examined each and every galaxy within the data set, that the classifications were of higher quality than could have been produced by a single expert looking at each data point. And so the effort through Galaxy Zoo resulted in the largest ever galaxy census and produced many novel discoveries and many publications. So it's been a huge contribution to the scientific literature. Interestingly, we've found through launching multiple Zooniverse projects that we also observe the identification of many unknowns within our data sets. So something that we find when working with members of the public via citizen science is that people get distracted. So quite often when looking at through the data, volunteers will go off task and find other things that are interesting in images. And one such serendipitous discovery was made by volunteer Hanny von Arkel. So shortly after Galaxy Zoo was launched, Hanny was inspired to contribute to Galaxy Zoo via one of our other contributors to the project. So Brian May of Queen fame was a contributor to Galaxy Zoo, and you can see that Hanny's got her guitar in this photo in the image here. And so she noticed in one of the images she was classifying that you can see below the image of Hanny, there's a green blob just underneath the galaxy, and it turned out that this image was effectively 1 in a million. It showed a phenomena that had previously been theorized, but there hadn't been any evidence to show at that point. And it was an ionized gas cloud, and I'm not an astronomer, but apparently the identification of this object is something that can be studied to help us learn more about the early universe and the evolution of galaxies. So it was a significant unknown found within the data set, and there have been many other examples like this that we've found through engaging members of the public with research. So the effectiveness of engaging the public in large-scale data analysis has resulted in many other teams, research teams, wishing to adopt a citizen science approach. And this has been because many other domains have experienced a similar increase in data production capacity. So we now have hundreds of different research projects on the Zooniverse platform, looking at all sorts of phenomena, from studying antibiotic resistance in TB through to examining penguin population change in ecology projects. We have many humanities projects, so involving the transcription of historical documents, And this is what the Zooniverse looks like as of yesterday. So I screen grabbed these statistics off of the Zooniverse front page yesterday. So as you can see from this, we now have more than 2.5 million registered volunteers on the platform. We have over 350 projects that we've launched on the Zooniverse. We've had over 700 million classifications across these projects. And this has resulted in much research, so many papers and many data sets, many analysis, and a lot of software that's been developed as well as part of those analyses. So how are we applying citizen science at the Crick? So in a similar origin story to many of the other research projects that have utilized the Zooniverse platform, the citizen science work at the Crick is a story that starts with too much data. So as I'm sure many of you are aware, there have been a lot of technological developments within electron microscopy, which mean that it's now possible to produce terabytes worth of data overnight. Thousands of images can be produced exploring the three-dimensional structure of cells. However, these technological developments have pushed us into a position where the research bottleneck has now shifted from data production through to data analysis, as the gold standard approach for analyzing this data typically remains manual segmentation by expert electron microscopists. As you can see, Anne, one of my colleagues in this image here, doing segmentation on a desk in the office. So it's a very slow manual process fully analyzing this data. So the team based at the Crick in the electron microscopy science technology platform had heard of the Zooniverse and reached out to myself and the Zooniverse team back in 2016 with their idea to apply citizen science for analyzing electron micrographs. And it was through this initial conversation that we decided that we would go ahead and we would give this a go and build a project to try and tackle the segmentation of electron micrographs. And this was the first time we'd built a project like this on the Zooniverse. There have been previous projects that had involved the analysis of micrographs, however, not the segmentation. So previous projects had looked at identifying single protein points within images, but we'd not previously had volunteers using a freehand segmentation tool on the platform. So a lot was involved in the development of this project. We had to develop the drawing tool in order to enable the task and figure out various problems like to how to appropriately display the micrographs while providing three-dimensional context information for the volunteers so that they could perform the task. In collaboration with the team at the Crick, we identified an appropriate data set for the analysis. We decided to look at HeLa cells because they're so commonly used in biomedical research. We wanted to make sure that the efforts of the contributors, the volunteers, would generate an analysis that would be as broadly useful as possible. And we decided to start with the segmentation of the nuclear envelope because we believed that it was likely to be one of the most simple organelles to segment. So it seemed a very sensible place to start. And it also mirrored what was quite often the process of the research teams in segmenting, that they would quite often start with the segmentation of this structure prior to segmenting other features within the cell. So we built the project, we launched it after a beta review process, and it ran for just over a year. We collected, I think, over 100,000 classifications from the volunteers over our data set. And after looking at this data, we were very pleased to find that despite it being a challenging task and despite there not being a precedent of other projects taking this approach on the platform, the volunteers were very good at doing segmentation. So if you click on these on the left-hand image, you'll see all of the segmentation submitted by volunteers for a single image within our data set that was uploaded to the project. And you'll see that although there's variability, most of the segmentations are very good. And the image on the left-hand side that you can see also speaks to this, that as you would expect from volunteer classifications, you can see that there is variability, that some people do segment other pixels that are quite far from the feature of interest. However, you can see from this image that when it comes to a consensus, when you look at all of the segmentations together, what we want, the segmentation of the nuclear envelope, emerges from the multiple classifications. And this brings me to the next slide, the point of aggregation. So one of the biggest challenges that we encountered with this project was how to make sense of the multiple segmentations per image. So we had to work quite carefully to figure out a way to form consensus, a single segmentation from the multiple segmentations that were submitted by volunteers for each slice of our data set. And we developed a new aggregation algorithm that we called CREA. And the headline finding from this slide is that it worked well. We were able to form an appropriate analysis pipeline that could take the volunteer segmentations and produce something that worked really nicely. So clicking through to the next slide, I think this is an embedded video, be able to play for you here. So you should be seeing, so these were the 18 nuclei within our volume, segmented. So all of these represent just pure volunteer effort. And as you can see, very high quality. We had some expert data, some expert regions of interest, and we found that the volunteer data compared very well to the expert segmentations that we had. However, even with volunteer help, we found, well, we're aware that the amount of data being produced by the microscopy community means that we need other approaches. We need to adopt machine learning algorithms into our pipelines. So we used the data that the volunteers produced, the aggregated segmentations, and we trained a convolutional neural network to perform the automatic segmentation of the nuclear envelope. And this performed very well. So I'll just toggle on to the next slide and video for you. So this shows in gold is the expert data, green is the volunteer data, and magenta is the convolutional neural network predicted segmentation for the nuclei for this particular cell, and you can see that they're all highly comparable. And moving on to the next video, this is just showing moving up through the volume so that you can see the two-dimensional segmentations for each individual slice. And again, you can see that the volunteer aggregations and the machine prediction were very comparable to the expert data. So to conclude, it worked well. And so, sorry, just waiting for this video to buffer. So the critical part of this research, so the real kind of headline finding of this project was that we were able to drastically reduce the amount of time taken to segment the nuclei within the data set. And we found, so it apparently would take a single expert approximately 30 hours of effort to segment a single nuclei. However, our predictive algorithm took only 1 minute. So with this approach, we were able to increase data analysis by a factor of about 1800. So the data analysis of our from our predictive algorithm to segment one nuclei was 1800 times faster than an expert. And we ran our model on other data sets as well, and we found that this model also performed well in novel contexts. So on data of higher resolution, data produced by different modalities, so FibSem, and we also found, surprisingly, that our model was able to perform well on a mitotic cell. And even though the nuclear envelope in this case, as you can see from this image, the nuclear envelope is slightly degraded, our model was still able to identify the nuclear envelope in this context. So the success of this project meant that we have taken this approach forward, and we're developing other projects. So these are a handful of the projects that we've had on the Zooniverse platform. A number of these are now complete, and we're in the process of analysing the data from these projects. We've run projects to look at mitochondria, the endoplasmic reticulum, and fat droplets. And these have been brought together under what's called an organisation. So this is just multiple similar projects on the Zooniverse platform. And I should say as well that we've got some new projects in the works, and we're looking to share these to our volunteer community for review very shortly. So this is a slight advance on what we've been doing previously. We'll be using correlative light images in conjunction with EM images to guide volunteer segmentation in this context, because we're now moving into structures that require markers to be unambiguously identified. So things like the lysosomes and the Golgi of the cell, which are very difficult to identify just from the electron micrographs alone. So it's exciting times in the Etchiverse. So this just brings me on to the final part of the talk. I appreciate that I'm close to the half hour now, so I'll go through this very quickly. So if this has inspired any of you to build your own Zooniverse project, it's straightforward. So a few years ago, the Zooniverse built a project builder interface that's free to use to generate your own citizen science project. It's available at this URL, accessible on the Zooniverse platform. There are three core components, I would say, to think about when you're building your project. You will need to think about the data that you want to share. So, and I should say that the Zooniverse is able to share multiple types of data with volunteers. So we can share images, we can share video, we can share audio, and we're also moving to a state of being able to show data to the volunteers in the form of interactable graphs. You will also need to think about the tasks, so the series of tasks that you will be asking the volunteers to do to generate the metadata that you require for your downstream analysis, and we're able to handle questions, drawings, texts, and surveys. And you'll also need to think about the community that you'd like to engage with via your project. And it's possible to share Zooniverse projects with members of the public openly on the Zooniverse platform, or privately with your own communities. So to get started with building your Zooniverse projects, you'll need to first register. That's the only prerequisite to build a project. Sign in and click Build a Project. So it's as simple as hitting that tab, create a new project, and then filling in some very basic information. This is all editable downstream, so you can change this before you launch your project. And there, just to draw your attention to two of the most important tabs on the project builder, the workflows and the subject sets. So all of these different fields on the left-hand side of the project builder page refer to various important components of building a project. However, I'd say these two are likely the most important. So subject sets is where you upload your data. There's a graphical user interface where you can just drag and drop. And there's also a Python client for more complex data uploads as well, I should say. For workflows, the workflow tab corresponds to the sequence of tasks that you would like the volunteers to perform. And as I've said, there are multiple task types, from asking questions such as which segmentation best highlights the boundary in the case of synaptic protein zoo, There are survey tasks. These are commonly used in ecology projects to populate with multiple options alongside images to help guide volunteers in the task. There's also transcription. This is commonly used in humanities-style projects to transcribe texts from pages. And there's also drawing tasks, which you'll be familiar with from this presentation, the freehand drawing task. And also there's the possibility of putting single points on images to identify things like the position of white blood cells in the case of Monkey Health Explorer. And yes, the segmentation of biological structures in HSL. And then if you want to share your project with the private community, you can go to town as much as you like with the various other features of a project, or more or less dependent on your own needs. However, if you wish to share your project with the Zooniverse community, then the other fields will need to be completed to a good standard. The project will be reviewed by members of the Zooniverse team and also the Zooniverse community before being approved for launch if the project reaches the appropriate standard. And as I say, you can share your project with your own private communities or the public. There can be different benefits to either approach that I won't go into depth about here. There are many other further resources out there. And if you are interested and you'd like to connect and learn more, I'm very happy to receive emails and connect. So do get in touch, and thank you for listening.
Thank you, Dr. Spiers, for your insightful presentation. It's very impressive how much you've been able to achieve by involving citizen science volunteers. We'll now start taking live questions for the webinar. If you have any questions you'd like to ask Dr. Spiers, please do so now by clicking on the Ask a Question box located on the far left side of your screen, and we'll answer as many questions as we have time for. So I have my first question here for you. Let's see. You touched upon this briefly. How do you get around inter-rater variability and signal-to-noise detection in your current study? What are some good measures that people can think about as they design their own studies?
Sorry, can you repeat the question?
Inter-rater variability, yes.
So I think some of the first things to think about when designing your study, you'll want to make sure that you're starting from a sensible point in terms of the quality of your data. So making sure that the data is as clear as possible so that the features of interest that you want to be annotated are as clear as possible. And also, so I didn't touch on this at all, but there are other mechanisms of making those features of interest clearer. So, for example, in the ER project, we used a label to make the endoplasmic reticulum clearer relative to the background. And similarly, with the correlative data that we're using within the projects to look at the lysosomes and Golgis, that's sort of a similar approach. So I think as a very sensible first step, just making sure that the data that you've got is as clear as possible and that the features that you're interested in are highlighted as clearly as possible is a very good place to start. And then ensuring that the instructions that you give are as clear as possible, providing good examples. So that's all very good practice within project building, making sure that you provide good tutorials, good field guides, good instructions for the volunteers so things are as clear as possible. And also there's a lot of nuance when it comes to selecting appropriate fields of view to make sure that you're focusing people's attention onto what's interesting. So in taking the nuclear envelope project, for example, we had, as you'll have gathered from the data that I showed, we had a very large volume that we were interested in, but there were multiple cells within that volume. So we made a decision to focus on the 18 nuclei that were selected as regions of interest, and we cropped those out, then made the subjects for each of those regions of interest so that we could focus attention onto smaller areas, so that there was less confusion between the different nuclei, or less possibility for confusion. So I think there's a lot to be said about good project design and simplicity, but then also you do have to have mechanisms in mind for the aggregation pathway down the line. So as I discussed, we had to develop a novel aggregation algorithm for the nuclear envelope, and that was partly because the freehand tool had never been used on this universe before. So there was a lot of work generating that tool so that we could reduce the noise between the volunteers and remove outlying data sensibly.
Awesome. Thank you very much. I have a next question here. What are some of the techniques to motivate citizen science volunteers to participate?
So I think in citizen science broadly, there are lots of different ways you can motivate people. One of the conversations that we have on sort of a 6-monthly basis or so within the Zooniverse team is the idea of gamification, but it's a concept that we tend to err away from on the Zooniverse side, because we find that people's intrinsic motivation to contribute to research, from wanting to help science or just pure interest in the subject matter, seems to be motivation enough for people to contribute. And we find that sometimes when you gamify, because some projects in the past have explored it tentatively in a minor way, and it's been found that it's led to unexpected consequences in terms of data quality. So I think we tend to just rely on people's motivation, but there are other things that you can do, and there are other things that other teams do.
Fantastic. Well, thank you very much for that, for your very thoughtful answers to the questions. Thank you again, Dr. Spiers, for your time today and your important research. Now we'll move on to our second speaker. Oh, sure. Now we'll move on to our second speaker, Dr. Martin Priessner. Dr. Priessner, you may now begin your presentation.
Thank you very much for having me and for giving me the opportunity to present the research that I hope that you can see my slides now. Yeah, thank you very much for having me and for giving me the opportunity to present to you the research that I've carried out during the time of my PhD, which I've carried out at Imperial College London, where I developed a tool which we call CAFI, which stands for Content-Aware Frame Interpolation. And during my talk, I will go through 4 parts of my presentation, where in the first part, I will explain a little bit more what the full title of our publication actually means. Then I will give you a brief introduction into the complications of fluorescence microscopy and how CAFI could be helpful for you to overcome some of those limitations. Then in part 3, I will share some of our own experimental results, which we have conducted and carried out using this tool. And in part 4, I will show you a brief demonstration of how you could use this tool for your own research. Okay. To the title, so content-aware frame interpolation. This basically means content-aware that we use data to we use a tool which learns from to learn pattern from data to basically perform the task of frame interpolation, which means if we have a sequence of microscopy images, that we increase the frame rate of those images, given, for example, 3 input frames, that the model learns to the patterns of those movements from one frame to the next to interpolate an intermediate frame between 2 consecutive images. So resulting out of 3 input frames, giving 5 output frames. Then deep learning based super resolution. I know I go a little bit into the foundations, but considering that it could be that there are a lot of people which have different backgrounds, I still explain it. Deep learning is basically a subpart of artificial intelligence and also a subcategory of machine learning. And to achieve basically the aim that we increase the temporal resolution, we use neural networks to basically achieve this task. And finally, we apply this tool, which we implemented on different bioimaging platforms, meaning on different microscopy modalities. So basically, why can CAFI be helpful for a microscopist? So especially when you do fluorescence microscopy with probably confocal microscopes, they often have the problem which is called the eternal triangle of compromise, which means that at a given resolution and speed and sensitivity, that you can just optimize or improve one parameter of those parameters by compromising one or the other. So for example, if you want to improve the resolution of your collected image, this means that the speed of collection will be reduced. Or for example, if you want to increase the speed of collecting the images, then the shutter, which allows the photons of the fluorescence to reach the detector, will be open for a shorter period of time, which then reduces the sensitivity and therefore the signal to noise ratio. Another challenge, which is often a problem for microscopists, is that it's called phototoxicity, which means that basically cells which are exposed to laser light for a longer period of time, the cells will become unhappy and eventually die, which means that also the scientists carrying out those experiments will be unhappy as well, because they cannot observe the biological processes that they are interested in. Another problem is also photo bleaching, which means that the fluorescent probe, which is taken up by the cell, and which basically causes the fluorescence emission after excitation of the laser light, gets weaker over time. So the longer you measure a cell given an exposure to the laser, the weaker the signal becomes and the less you can see actually the labeled compound of interest. And all those limitations become even more pronounced and more limiting when you look into multicolor imaging or 3 or 4D imaging processes, which take a longer period of time and therefore make those problems even more pronounced. Some ways of overcoming these challenges could be that to basically get better hardware, but this obviously at the cost, and sometimes it's intrinsically not possible because of intrinsic limitations of the hardware. Another solution would be to use better fluorophores, but this comes with the same limitations that they are just sometimes not available, and the synthetic efforts in designing new probes is actually really a difficult task, considering the synthetic routes and the fluorophores of interest, the uptake inside the cell, and there are a lot of aspects which need to be considered. And so the approach which we propose is to basically use AI and an AI tool in a post-processing imaging step, which can improve the situation in this case. And our research is built up on 2 fantastic publications which have been carried out prior, which one of them was carried out by Wen Bao while he was working at Google, who implemented the depth-aware frame interpolation tool, and Xiaoyu Chang implemented the zooming slow-mo network, which both of them are capable of interpolating images along the time axis, in this case of video frames, of which the zooming slow-mo network is also capable of upscaling the image and improving the image resolution. So what we did in our research for the implementation of CAFI is that we basically implemented the two networks, of which we took the depth-aware frame interpolation as it is, and we changed the structure of the zooming network that it can perform the interpolation step without any upscaling capabilities. But we also kept this option as well, in case if people are interested in exploring this functionality as well. Okay, so what are the actual capabilities of CAFI? So the basic one is, as I described before, that you have a microscopy sequence, maybe some cells moving over time. And basically by applying CAFI on your data set, which you have, you can improve the image density of the image sequence which you collected. And by iteratively applying the same algorithm or the same tool on iteratively on the same data set allows you to even further improve the density of the image sequence. This is one option. Another option, which we also explored, was to basically use the same tool along the set dimension of the data. So if you have set stacks where you record cells in different depth dimensions, you can use the same tool to improve the density of information in this dimension as well. Or you can even combine both of the functionalities to basically perform interpolation step along the t dimension and the set dimension, improving the amount of information generated by the AI, basically. Obviously, the last step is a very challenging one because it assumes that the interpolation steps which have been carried out by the time dimension is accurate. And if there are some artifacts which are created, then they are like more pronounced in the second iteration step. So this needs to be taken with caution when applying this kind of methodology to your data. And so how did we evaluate actually CAFI's performance on our own data set? So the basic methodology was that we have a sequence of some microscopy images where we basically have a ground truth sequence where we remove every second image. And then in an interpolation step, we interpolate the missing frames back into the sequence and then compare basically the ground truth sequence with the CAFI interpolated sequence. And in order to find out how well it performed, we evaluated the resulting output with 3 different evaluation metrics, one being the root mean square error and the peak signal to noise ratio, which are both pixel-based evaluation metrics. And we also evaluated it on the structural similarity metrics, which is a more structural-based similarity metrics commonly used in computer vision tasks. And we compared basically the output of the CAFI networks with 3 traditional ways of performing temporal interpolation, which is bicubic and bilinear interpolation, and none is basically just a simple frame duplication to see how much the network actually improves over a simple comparison of the input side. Now I'll show you an example where we applied basically this methodology on one data set. This is a mitochondrial data set, where in the left upper corner you see the input, on the right upper corner you see the ground truth of the data set, and in the middle you see the CAFI interpolated sequence. And we performed it basically with one-time temporal upscaling, and in the second time we performed that upscaling twice. You can see that the overall movement looks much smoother in comparison to the input sequence because it contains much more frames, obviously. But on the other hand, you can also see that and I will restart the video just to show you this again. But you can also see that the randomness of the movement from the ground truth gets lost in this process, because this is something which is intrinsically not possible for the network to pick up in this example. And in the lower parts, you see actually the comparisons between the ground truth and the input, and ground truth to the CAFE interpolated output. Okay. Now looking at the numbers of the similarity metrics, which I described to you before, we can see that basically the traditional interpolation techniques are performing worse in comparison to the CAFE networks. So, and already the pre-trained network, which has not been fine-tuned on any of the data, is already outperforming the traditional mathematical approaches. And when after fine-tuning the networks, you can see that the performance even increases even further. The next we actually tried to use a more systematic approach to evaluate this network, and this was by basically choosing a simulated data set where we could control each parameter, like the size of the dots, the speed in which the dots were moving around, the random direction changes, and a lot of other parameters. And we applied basically the same methodology to this kind of experimental setup. Again, here you can see, again, left upper corner is the input frames, right upper corner is the ground truth, and in the center you can see in this case, the Dyn interpolated images. And now with 4 times upscaling along the time dimension, you can see that also the randomness of the movement gets lost, but the overall direction of the frames are reproduced actually quite nicely. Then again, looking at the evaluation metrics. So in this case, we actually performed the experiment also checking how well the network performs different movement speeds of those dots. And so we can see that in both cases, like for slow and fast movements, both networks within our CAFE framework perform better than the traditional approaches. But we can see that with increasing particle speeds, the performance also drops, which is understandable because the quicker the particles move, the more difficult it is actually also for the net to pick up the direction and the overall speed of the particles. And if you look on some of the arrows in the lower part of the image, you can see at a higher velocity and higher particle speeds, the networks start making mistakes, which is something that everybody who is using such tools needs to be aware of. For example, Dyn is basically when it's not sure in which direction the particle is moving because of the very high speed, then it starts interpolating the images into 2 different directions where both of them could be a possible solution. On the other hand, assuming slow-mo actually doesn't interpolate the points which it doesn't know where to put them. And so those are basically artifacts which need to be considered when you do downstream analysis in a later stage. Okay. I just realized I forgot to push the slides, but I hope now you can see it. So basically, the left panel shows again the performance of the networks in comparison to the traditional methods, where the CAFE networks perform better than the traditional ones. And the side in the pictures, you can see some of the artifacts which are created when going to very high velocities. Next, we actually tried to evaluate the network also on its ability to improve the tracking results of those particles. So using this simulated data set gave us the possibility to basically use the ground truth tracks of those particles and compare it then in the end with the particles of the interpolated networks. And as mentioned before, you can see that the ground truth particle movements are actually a little bit more random, which means that they have like some Brownian motion, which the Dane and the ZS network cannot like correctly reproduce, but the overall trends are fairly similar. And when looking at the results, so we basically applied the 2014 ISBD particle tracking challenge metrics onto the performance which we onto the analysis which we carried out on the simulated data set. And we can see that again, like ZS and Dane performed better than non and bilinear interpolation. And interestingly, bilinear performed worse in comparison to simple frame duplication, because bilinear interpolation basically linearly blends together two consecutive frames, producing a lot of artifacts, which obviously the tracking algorithm has troubles with coping with it. And we also performed the same experiment then on on real lysosomal data, where we had the problem that we didn't really have the ground truth tracks, but for like simplifying this problem, we basically took the ground truth tracks from the ground truth sequence and compared basically the interpolated results with the ground truth tracking results as a comparison. And also in this case, we can see that specifically zooming slow-mo performed better than Dane and Dane. Both of them performed better than the more classical interpolation results. Now we can also look into one example. Zoomed in section where you can see, for example, where some of the lysosomes which are moving around will be missed from basically the non-interpolated images. Yeah. Okay. And now I show, yeah, because of interest of time, I probably just show you one more example of a data set which where we also applied CAFI to. This was a quite challenging one because there's a lot of movement happening within. Just showing you this again because it's quite quick and short. But in the left lower corner, you see the input image, and in the right side, you see the two interpolation results of CAFI. And now I just briefly show you like a demonstration, a pre-recorded demonstration of how you could use this tool for your own project, basically by using a Google Colab notebook. So basically the tool is implemented in Google Colab, which is an open source or like openly available source from Google, and it's divided in different sections where you find a lot of information of how to use the notebook and how to basically operate it. And first you need to connect it to the cloud server because it will use basically a GPU which is provided by Google. After running this cell, you will see whether you have a GPU available or not. Then if you don't have it, you can go to those settings and select the GPU in case if it isn't connected. Then you connect it to your Google Cloud storage, in this case, Google Drive. So after copying the registration key, there will be a folder showing up on the left-hand side where you have access to all the data which is available on Google Drive. And this is to upload also for your images. Then by running this cell in section 2, you install all the necessary packages which are necessary for running this notebook. And there you find also, there will be a new folder appearing calling Zoom Interpolation, where you have a demo folder with some examples. Then for doing your own training, you can select your own training data, which will be processed by the notebook. It's recommended that for bigger data sets, which are bigger than 10 GB, it's recommended to use the scripts provided and do it offline because Google Colab has also some limitations in the compute performance. But for smaller data sets, it's absolutely possible to run it within the notebook. And so you can just select the data you want to process and add the by right-clicking, you select the path, and then you select also the image size. And then after running the cell, there will be 2 new folders appearing, which you can then select for actually running the training. And for the training, you again need to select those 2 preparation folders, which have been prepared in the previous step. And you need to select also whether you want to have any zooming capabilities, and also the input dimensions of the images need to be provided. Then also how many iterations you want to train the network for. How often should the checkpoints be saved? Because there's the option to save also the intermediate results of the network, and the learning rate needs to be selected, and so on. And the warm-up iter basically tells you how many iteration it takes for the learning rate to reach its full height, basically. And after you have specified the parameters, you can just run the cell, and it will load basically the data which you've processed before and runs the fine-tuning. After this has finished, you can also visualize those results to see how well the model has. You then also upload the new model to the software that you will, or to the tool basically, that it knows that it will use the pre-trained model, and then it basically, there you show basically just the learning effect of the fine-tuning. In this case, it's just a demonstration. And then you can move on to section 5, where you can actually perform the interpolation on your own data. So you select again from your Google Drive folder, the folder where the images are located. You select the image dimension of how big the image is. You select in which dimension you want to interpolate this image, in T or Z dimension. And then you're basically ready to go. And then you also select the location where the output should be stored or saved. It's very recommendable to choose a folder inside your Google Drive, because if the notebook disconnects, then the data would be lost. But usually this is a standard setup. Then after running the cell, it basically will use the folder that you provided with the data and creates you a new folder with the interpolated images, which you then can download basically for your own inspection. So by just right-clicking, it's like in the Explorer, you can just right-click and press download, and then it will download the file onto your computer. Or otherwise, if it's stored on your Google Drive, you can just download it on your Google Drive via the browser. In the last section, there are some more details on how you can evaluate the images, which I won't go into more details because of the interest of time. But if you're interested in how to use it, there's a more detailed explanation video online on my GitHub repository. And if you're interested, I highly recommend you can watch that and go through the whole methodology slower, step by step. And in general, I would recommend people who are interested in using it to first use it with our provided pre-trained networks, pre-trained models. Then you see whether the results are actually already looking promising. If they do so, then you can go ahead and fine-tune the network. And the fine-tuning step is actually something which is sometimes a little bit like tricky to do, because you need to set the parameters right, the learning rate, and so on. So it will take some time to get it correct. That's why I would recommend using it in the first place with our pre-trained networks, to not waste your time, basically. And now I almost reached the end of my presentation, where I want to point you to the direction of where you can find this tool. So it's basically hosted by the zero-cost deep learning for microscopy platform, which has a lot of other very interesting tools, deep learning tools for microscopy, which you can use, starting from nuclear segmentation, denoising, image upscaling, artificial labeling, and they all follow basically the same pipeline of data processing. You can upload files on Google Drive, you can process and fine-tune the network using the Google Colab environment, then you can perform the inference, producing a new output, and then you analyze the data on your own computer. And there are a lot of helpful tutorials which you can also watch, which will help you to basically get along. And with this, I reached the end of my presentation. Yeah, and the final word of caution is that everybody knows that deep learning and AI can produce magical, really nice results, but it's always important to, whenever you use those tools, that you evaluate the models on your output and check whether the results are actually what you expect them to do and so on. So always use those tools with caution. And now I reached the end of the presentation, where I want to thank all the collaborators which helped me to perform this kind of research, with a special highlight to Romain Lane, which stepped in as an additional supervisor, since my background is actually more from the chemistry side. And I want to thank also my supervisors, which allowed me to explore this area of research during my PhD, and also the funding bodies basically paying for all my fun research that I was allowed to do. And yeah, thank you for your attention.
Thank you, Dr. Priessner, for your insightful presentation. We'll now start the Q&A portion of the webinar for either speaker. In fact, if you missed your opportunity to ask Dr. Spiers, if you have a question to ask, please do so now by clicking on the Ask a Question box located on the far left side of your screen. So we have a first question for you, Dr. Priessner. With the explosion of the number of available deep learning algorithms such as CAFE, what do you think an average biologist can do to get started with AI and deep learning?
That's a very good question. I think nowadays we already reached a point that not so much coding skills are actually necessary to really use deep learning algorithms. There are a lot of tools out there which make it very easy for microscopists and biologists which are not fluent, for example, in Python or in any other programming language, to actually just go online and use those tools. One platform which I've highlighted is basically this zero cost for DL for microscopy platform, which is basically a no-code solution for this kind of task. So you can actually, without any coding skills, use those kind of tools from your browser, and it makes it quite easy. And there are a lot of other tools available out there already integrated in Fiji or in IC and some other imaging processing platforms. And I think in the future this will be even more common to be integrated in normal image analysis pipelines. And so, yeah, I think just watch out for those solutions which are out there, and don't be afraid to step in and try to use them, even though it sounds like you need to know coding, but it's sometimes easier than you think. Yeah.
Thank you very much. The next question is, in deep learning, what are some measures that can be implemented to avoid or minimize artifacts?
This, I guess, is a question which everybody would like to know in very detail, but it's a very difficult question to really answer in great precision, because it always depends on the task that you're looking at. So with image processing, obviously you have nowadays quite easy ways of collecting a lot of images. So, and usually what's in the field of computer vision or in the field of deep learning is that the more data you have, the better the results are. So, but even if, like, because you can train the network on bigger data sets, and then it learns to generalize the better to the data that it's trained on. Other ways of like overcoming this challenge might be, if you have like limited data available for your training, that you can use some data augmentation methods, where you basically flip and rotate and crop images to basically augment your data set, which increases the amount of data that you have available for training. But finally, it always comes down to the person carrying out the research to basically see whether the results from the network are reflected correctly in what you expect to see. And so it's always important to perform some downstream analysis on the data that you are applying the network on, to make sure that no artifacts get created, or the artifacts that are created are not influencing your research.
Thank you very much. I know we've gone over time a little bit. There's one other question that I'd like your thoughts on. Some researchers are skeptical about reproducibility about AI-assisted or generated results, especially when it comes to predictive models. What can users to improve reproducibility when it comes to deep learning models such as CAFE?
Yeah, so reproducibility is a common issue for deep learning as well, because deep learning is seen as this kind of black box which does something and nobody really knows what's happening underneath. But in the end, what is important is that whoever uses CAFE or any other deep learning tool is that they provide the pre-trained models for other people to reproduce, to be able to reproduce what they have observed. And another thing what people need to pay attention to is that also those pre-trained models can have some problems intrinsically in the training. That, for example, if some training or some testing data gets into the training, it would basically teach the neural network to memorize patterns from the results that it's actually about to predict, and then it's artificially increasing the performance, which is actually not the case. So it's important to then also provide the training data to the users that they can actually really reproduce the training it themselves as well, to make sure that this is not artificially improved by maybe some mishandling of the data sets.
Fantastic answer. Thank you so much. Thank you again to both of you, Dr. Priessner and Dr. Spiers, for your time today and your important research. That concludes our presentation. On behalf of Leica Microsystems, we'd like to thank LabRoots for underwriting today's educational webcast. Before we go, I'd like to again thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provide at the time of registration. The webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay, and we encourage you to share that email with your colleagues who may be interested in this topic of AI microscopy. Until next time, take care, everyone. Thank you so much, and goodbye.
Thank you.